Mucor introduction. Mucor is a saprotrophic fungus. The appearance of the sporangiophores and sporangia resembles a collection of pins, and hence it is commonly known as pin mold. As the matured sporangia are black in color, it is also called black bread mold. Occurrence. There are more than 50 species that have been reported in this genus. It grows on dung, wet shoes, stale moist bread, rotten fruit, decaying vegetables and other stale organic media. It can be grown easily in the laboratory on a piece of moist bread or on horse dung kept under a bell jar in a warm place for 3 or 4 days. Somatic structure. The body of mucor is composed of a mass of white, delicate, cottony threads collectively known as mycelium. It is always much branched and coenocytic, i.e. aseptate and multinucleate. Each thread of the mycelium is known as hypha, which is aseptate. The mycelium spreads in all directions over the substratum. Some of the hyphal branches penetrate down into the substratum to absorb water and nutrients. Reproduction. This takes place vegetatively, asexually and sexually. The asexual reproduction by formation of spores is very common in mucor. Vegetative reproduction, fragmentation of the mycelium, results in the multiplication of the fungus by developing fresh stock of mycelia. Asexual reproduction. Asexual reproduction is by sporangiospores or chlamydospores, sometimes by oidea. Sporangiospores or spores. Mucor readily reproduces asexually by forming sporangiospores or spores. Here the spores are produced in sporangia. Under favorable conditions of moisture and temperature, numerous slender, erect hyphae called sporangiophores arise from the mycelium. These sporangiophores are hyaline and do not produce rhizoids at their base. The apex of the sporangiophore enlarges to form a vesicle which may be globose or spherical, multispored and columellate structure, the sporangium. As the hypha begins to swell, the protoplasmic contents migrate to its tip and accumulate there. The protoplasm differentiates into a central vacuolated region and an outer fertile dense peripheral region containing large number of nuclei. The central vacuolated region is separated from the peripheral region by a cleft or furrow, and this furrow later develops into a wall. This central sterile region is called columella. The peripheral fertile region forms spores. The spore wall thickens and darkens. The wall of the sporangium is thin and brittle. The columella swells due to the accumulation of a quantity of fluid and exerts a considerable pressure on the wall of the sporangium. As a consequence, the sporangium bursts, setting free the spores. The spores are dispersed by wind, and under favorable conditions, they germinate in a suitable medium to produce a new thallus of mucor. Chlamydospores. It is sometimes seen that under unfavorable conditions, the mycelium of mucor becomes segmented into a short chain of cells. These cells swell up and become thick-walled and large. These are called chlamydospores. These spores are resting spores. They germinate under favorable conditions to give rise to somatic structure of the fungus. Oidea. Sometimes the hyphae produce yeast-like budding cells called oidea. Sexual reproduction. The mycelium of most of the species of mucor is unisexual and hence unable to produce both the male and female gametangia. Sexual reproduction takes place when hyphae derived from different spores come in contact with one another. There is no morphological distinction between the two sexual hyphae, although physiologically they are dissimilar. Since physiologically dissimilar thyli, hyphae, are involved in sexual reproduction, this phenomenon is called heterotolism. The two sexual hyphae are called plus and minus strains. Here plus strain and strain hyphae readily mate and hence are called mating types. Whereas plus and strains do not mate. The growth of the plus strain female is more vigorous than that of strained male. In heterotolic species, sexual reproduction begins when plus and strain hyphae intermix. In some species, any two hyphae from the same mycelium derived from a single spore may initiate sexual reproduction. This phenomenon is called homotolism. When a plus strain and strain hyphae of heterotolic species come in contact with one another, the tips of both hyphae swell to form progametangia. The two progametangia meet and adhere together at their tips. Each progametangium gets transformed into a terminal gametangium and a basal suspensor. Each multinucleate gametangium is regarded as a coenogamete. The wall between the two gametangia dissolves and the protoplasm of both gametangia fuse to form a black thick-walled zygospore. As the thick-walled zygospore has reserve food material, it is able to tide over unfavorable conditions. During favorable conditions, the zygospore germinates and undergoes meiosis to produce a sporangiophore and a sporangium at its tip. Such a sporangium lacks columella and produces numerous haploid spores. The spores are released due to dehiscence of sporangium. Upon germination, the spores produce plus, or, strain hyphae.